Alright guys, just want to get you started with a little bit of your information today. I do apologize for not being here, and please know that this is available on the YouTube webpage, so you can view this at home. Please also ask the sub if you need something paused because I'm moving too fast. You can write that down. I just ask that you are courteous to those around you, and let's try to keep the talking during this point to a minimum. You will have some time to work in small groups a little later today. So we've kind of been looking at how do we tackle the whole issue of this activation energy. And we said that enzymes can help lower that activation energy, which essentially will help reactions speed up. So kind of what I want to start off with today is, well, why couldn't we just heat up whatever we need? We know that heat will help chemical reactions move faster. We're adding kinetic energy. So why wouldn't that work? Well, we're going to talk today about some environmental things that can happen with enzymes, and one of those is our living systems are very sensitive to temperature changes, and we don't have a whole lot of flexibility with that. So again, we got to rely on enzymes, which are proteins that will increase the rate of reactions. We don't have to worry anything about temperature. Very rarely is there heat involved with this. They're going to lower the activation energy, what it takes to break apart or to put molecules together. And basically what they do is they make a little shortcut. They make a new reaction pathway. And that's where we're going to end up with today, is looking at how this feeds into some of our feedback mechanisms. Alright, so in your packets that I handed you about enzymes, you've got this picture, and I just wanted to show this because I feel here where I've got the blue star, this is probably where many of you think enzymes lie. You've got this little blob thing here. In our lab, we're using catalase. Remember, ASE is a common ending for enzymes. And we've got a substrate up here called hydrogen peroxide, something that's toxic that's produced in your cells. And we're going to break that down into two products. We're going to break that down into water. Hey, we'll deal with some water in our cells. That's no big deal. And oxygen. No problem there. Well, the other issue to this is sometimes enzymes will take two substrates. Over here, we're only dealing with one substrate, and they'll actually help facilitate and put that together into making one product. Over here, we had two products. So just something to keep in mind. This would still be an example of a chemical reaction. We talk about this in chemistry as decomposition, and this would be synthesis. So just something to keep in mind with our biochemistry. The other pictures that you'd reference, this is always what I show the little kids. You know, I try to keep it very simple for them and tell them, look here, look at the substrate. It's got a pointy part, and oh my goodness, the enzyme's got a pointy part too. And then it's got a little blunt part here, and oh, you've got to be kidding me, there's a blunt part there too. These just all fit together. And so I try to teach this, you know, as far as active sites and things like that go with them. Well, yes, we call this the lock and key hypothesis, and for the most part, sometimes that works. But what is kind of new to this idea, and what might be new to you for AP Bio, is something called the induced fit model. And I'll talk a little bit more about this. It's not exactly like the lock and key, and it kind of helps us to explain some of the unusual enzymes that exist in life. Alright, so let's kind of start off with the little kids idea. This is probably what I teach them, and what I'm teaching them right now is that the enzyme is specific to its substrate. So, for example, if this is catalase, are you sick about talking about catalase yet? Catalase is only going to break down hydrogen peroxide. If I have another toxin that builds up in my body, catalase isn't going to work with that. It doesn't recognize that other shape. And shape kind of plays a big role to this because just like you have keys to get into your house, hopefully they let you get into your home and not your neighbor's home. Or the key that gets into your car lets you get into your car, but not the person parked next to you's car. So that's kind of where we come up with this lock and key hypothesis. The fit between the substrate and the active site of an enzyme is exact, just like a key fitting into a lock. So in that analogy, the enzyme is the key, the substrate's the lock. We do have a temporary point where those two are joined together. That's called the enzyme substrate complex. I don't think that's a huge phrase that you should know. You can pretty much say complex, put two things together, enzyme substrate. Oh, must be when the enzyme and substrate are together. And then what's formed are products that have a completely different shape from the substrate, so they really don't have any conformation changes or anything like that to the enzyme. So the enzyme's done with it, releases it, and that enzyme's active site is then again available for something else. So here's what it looks like in picture form. You've got your enzyme in the yellow, substrate in the pink. Again, this graph should look familiar. We talked about this with some of our energy problems from before. And this, without an enzyme, your activation energy that you would need would probably be something like that. So the enzyme's lowering that. And you end up with your products down here. So you start with kind of your reactants and end up with your products. So here's the key thing to kind of know from this. Your enzyme can be used again over and over and over again. So that's great. Your body is all about conserving and reusing things that it gets. And this is an example of how that works. So what's a little weird and kind of the new part to this is the whole induced fit hypothesis. So what we know by having a little better understanding of biochemistry is we've got some proteins that can change their shape. And so when the substrate combines with the enzyme, that substrate actually induces the enzyme to change. It's kind of like if you hold your hand out to shake someone's hand, you kind of wrap your hand around theirs unless you do the whole dead fish handshake. So the active site on that enzyme's molded, kind of like if you put a latex glove over your hands and you were wearing a ring, that glove doesn't just stop at the ring, it kind of molds itself over top of it. Now, in this whole molding process, that's what puts the stress on the bonds of the substrate, and they're bent, or sometimes they're described as being twisted, and that's what makes your reaction speed up a little faster. So how we came up with this idea is we saw a couple groups of enzymes, and I'll give you some examples when we talk in class, who would react with a whole range of substrates. And that did not follow our lock and key hypothesis at all. So as we're learning a little more about molecular shapes, we find that some enzymes will act on similar molecules, not just specific ones. And this is kind of a huge breakthrough in understanding a lot of things about our bodies. Why do certain cells die when they do? Or why do some cells never die? And it depends on how these enzymes are reading different chemical signals. Also plays a huge role in how we get medicines to work. If you can have a medicine that mimics a molecule that your body produces, and you can get that medicine to match as closely as the family of molecules that you need it to, then that medicine can be added to your body if it's something you're not naturally making, and the enzyme should recognize it and do its thing. So enzymes really do play a key role in pretty much everything we're going to talk about from here on in the future. Alright, so what can we do to kind of alter the whole enzyme reaction? And there's about 5 ways to talk about that. The environment's probably the biggest one. Those are things we're going to explore in our lab. The rest are just kind of little bullet points to add on to this. I also think that this is information that you've had from honors before, so I didn't want to spend too much time with it. But let's kind of just talk about what we know about reactions. So this has no enzyme to it, non-enzyme reaction. So let's say if it would increase a substrate, increase a certain chemical, and that increase in the chemical is going to cause the reaction to increase too. You'd have this nice straight line. So the velocity or the speed of the reaction is equal to your reaction rate, and you're going to be calculating some reaction rates in your lab. Okay, so now what is the issue if we add an enzyme to it? Well, we see that you're going to, let's say we start at 0 here, and I'll put my first little tag right here that we're going to look at, and then we're going to look at the second point right here, and then I'm going to come way out here and look at a third point. Well, we see if I have this amount of substrate from point 0 to here, oh yeah, well that number's increased. Okay, so my reaction's increased. And then let's say I put, this amount of an enzyme in here, and I follow that up, and oh, by golly, we had another increase in our reaction rate. So isn't more better? Isn't that our idea? Well, maybe not so much, because look what happens out here at 3. At 3, I've got a leveling off point. I could even come out here and add something else at 4, but what I'm going to find occurring is I'm going to be at this very same point, clear out here. I've reached my V max, my maximum velocity. So this kind of should look like carrying capacity. Remember when we were finding the value K for a population? And we saw the populations could level out. Well, what happens with this model, this particular model we're looking at here, your substrate, let's say we're dealing again with hydrogen peroxide, and we've got those little molecules floating around. Well, we did nothing to increase our enzyme level. So let's say for every 1 hydrogen peroxide, I have a catalase molecule that's looking around. So those will be my little red ones. Now what you're going to find is there's going to be a point when every single catalase molecule that you have available for this reaction to occur is going to be occupied. The active site's going to have a hydrogen peroxide in it. And until that's broken down and that active site becomes open again, you're not going to have any other way of breaking down that substrate any faster. Basically what happens is when you get to this V max point, all of your enzymes are occupied. They're all busy. And you reach kind of a standstill. The enzyme's still working, the reactions are still going on, but that velocity has leveled off. So to the environment, you could change your amount of substrate. You could also change the amount of your enzyme. You would have to keep a constant connection. If you wanted this line to continue to go up like this, you're also going to have to show an increase in your enzyme. And we'll learn some pathways about how these enzymes are made. Sometimes that's not always possible. Sometimes your body actually gets backed up in these reactions, and you're at a standstill. So other things environmentally wise that you can do to change enzyme working conditions is you can either increase or decrease temperatures. And I've got an example up here of typical human enzyme. If we follow this down, I'd say you're probably about 37 degrees Celsius. That's usually your body temperature. And so we talked a little bit about if you would have a fever, if you would increase that, what you actually begin to do is you're denaturing enzymes and you're denaturing proteins. Your body's goal is to denature whatever's invading you, and that works usually pretty well. It's no big deal to have a fever for a day or two. But beyond that point, you're exposing your body to some pretty harmful conditions, and you're going to start denaturing good things that you've got going on, good proteins that you need. So what happens when you denature an enzyme is you mess up those hydrogen and ionic bonds. And so basically what happens is the active site, the shape changes. And so that causes a problem, because if the active sites change to the point where your substrate can no longer bond, then you're not going to have any enzyme activity going on. These temperatures, though, where enzymes work at are specific to the organism. We've got some bacteria that are at deep sea vents where you've got a lot of warm water coming up. You figure 100 degrees Celsius is where water is boiling. This is quite a bit different than a human body. And these enzymes are fine. They function perfectly there. And there's some evolutionary relationships. What caused those enzymes to be able to withstand? What bonds do they have? And ultimately that goes back to the organism's DNA. Alright, the other thing we'll look at is a change in pH, and we'll model this in our lab too. I pulled up the pepsin, the enzyme here, pepsin, because that's something that works in your stomach. And I guess you could maybe have an idea as to what stomach acid's pH is. It's right here at 2. Pepsin helps to break down some proteins as you digest. But not everything works best at that temperature. Another enzyme called trypsin, it likes to be a little more on the basic end. And so some enzymes like high pH, some like low. It just depends on maybe where they're at in the body or their function in the body. When you go to extremes, like let's say we take pepsin and we make it go a little more on the neutral side, towards this way, towards those extremes, the bonds that form pepsin get messed up. Specifically, you're looking at ionic bonding and a little bit of the hydrophobic interactions too. So again, it kind of goes back to your protein structure, which we learned about earlier. So those are kind of the 3 things. You can look at concentration of your enzyme, temperature, and pH. Those would all be a part of environmental conditions. Other things that you may find that change enzymes, something called cofactors. A lot of times these are metals, and if you look at the bottles of your vitamins, iron, zinc, copper, these are all important things to have. So a cofactor, it's a non-protein helper, just helps catalytic activity. We're going to talk about magnesium and how that helps photosynthesis occur. So it's not just that humans need these, other organisms need some of these metals also. If you have something that's organic, meaning it has carbon in it, that can affect your catalytic activity. Vitamins are an example of that. Vitamin C, for example, can help speed up different reactions. And then we kind of get into the examples too of, well, how do you regulate all this? And we're going to get into our inhibitors and into our competition part of this. So one of the things that you can have is something that's called a competitive inhibitor. It's going to have almost the same exact shape as a substrate, and what it does is it just kind of comes in, it hangs out in the active site, and it pretty much just blocks the competitive inhibitor from being able to bind. So it's something that looks kind of similar to your substrate, and it's not going to be broken down by the enzyme though, it just kind of occupies that seat. We call this competition. When we start to begin to look at cell communication, this starts to play a role. The other thing that you can have going on is something that's called allosteric inhibition. What happens is you get something that's called a non-competitive inhibitor. So it doesn't attach at the active site, it attaches at another location along the enzyme. But when it does attach there, it changes this enzyme's active site's shape. It's different from denaturing and changing the shape, but it's actually a chemical reaction that goes on. And we call this allosteric inhibition. So where we're kind of going with this, where these 2 rules go into in our last topic for today, talks about feedback inhibition. How do we build a chain of enzymes that will actually switch on and switch off some of the reactions that we need going on? And feedback inhibition plays into pretty much everything we're going to get onto from here on out with our talks. Today is just a introduction. But let's say we've kind of got our initial substrate up here at number 1. That initial substrate bonds to an active site, and it causes a whole cascade of effects. We've got all kinds of enzymes involved that break products down and continue to break products down and blah, 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 until we get this little end part right here. Well, what you find is once you build up enough of this little end part, it will become an inhibitor. It will bond to that enzyme and stop the whole cascade of effects that produced it in the beginning. And so this is your body's way of building in checks and balances. Once you've got enough of something, your body will shut the reaction down. If you need some more of something else, it's also a way of turning on a reaction to occur. And that's where this feedback inhibition comes into play. And again, it's kind of a part of everything that we talk about in the future. All right, so again, let's just think about this. I'm going to see you again on Wednesday the 28th. I want you to enjoy your Thanksgiving, have a nice break, work on all of your enzyme stuff in your packets. On the 28th, I really think we need to come in and finish up our lab on that day, and then we'll look at having our test over cells, cellular transport, and enzymes on the 30th, and that's a Friday. Okay, I hope you have a nice Thanksgiving and safe Thanksgiving and enjoy the time with your family.